Hi class, in this recording we're going to talk about tissue growth, stem cells, and tissue regeneration. Let's dive into it. So when we look at the tissue growth, this idea of growth usually is going to be when we're younger, increasing the number of cells. Once we're adults, though, we usually are going to increase the size of our existing cells. So instead of adding to our count, we just make them larger. The idea of having more cells generated is known as hyperplasia, and this is very healthy and normal for young children and people who are not done growing. So typically before the age of 21, hyperplasia is pretty normal. Once you hit the age of 21, you shouldn't necessarily be adding a lot more cells to your body, though, and that you can still have hypertrophy. And this is going to be the enlargement or the increase in volume or increase in size of current cells within your body. So when we think of this would be hypertrophy of the muscles. Like, most people want a little hypertrophy of their muscles. However, there's also going to be hypertrophy of the adipocytes or the adipose tissue as well. That can happen as we age. When we look at the growth of a tumor, neo means new, and plasia can refer to a tissue. So a neoplasia can be the development of a new tissue or a new tumor. And in this case, we call that a neoplasm. Now, we can have benign tumors that are not spreading, or we can have malignant tumors. When we say malignant, that means that they're actively spreading throughout the body. And the thing that makes a tumor is that the tissue is abnormal or non-functional. So it's still human tissue. It's still you. It still has your genetic code. The big issue is that it doesn't actually help your body. It just sucks up resources and doesn't contribute to the health of you as an individual or as an organism. Now, as tissues are developing, tissues will change over time. We can have the cells within the tissue differentiate. And they typically, when we have differentiation, we go from more general to more specialized. So from general to specialized. So we can have an embryonic tissue become something like cartilage or bone. Now, as this process occurs, most of our differentiation is going to occur in utero, but there still is some differentiation that occurs throughout our life cycle. For instance, let's say you skin your knee or you get a cut, you need to have some tissue regeneration to heal from that wound. We also have some changes to our tissue that occur without the stem cell being utilized. And this is known as metaplasia. And this does not utilize stem cells, but instead take one mature tissue type and convert it into another mature tissue type. So to really just zero in on the distinctions between these two. When we have differentiation, we go from a general tissue to a specific tissue. When we have metaplasia, we go from one specific tissue to a different specific tissue. A classic example of metaplasia is when we look at the lining of the vaginal canal. In prepubescent girls, it's a simple cuboidal epithelium. After puberty, it transitions to a non-keratinized stratified squamous epithelium, because theoretically after puberty, the lining of the vagina needs to be able to resist some abrasional forces. Another example of metaplasia would be the lining of the airways or the passageways of your lungs. And normally what we have is a ciliated pseudostratified columnar epithelium. Then it moves lots of mucus up and out of the lungs. For individuals that spend a lot of time smoking, these pseudostratified columnar cells will transition into stratified squamous cells. And when this happens, they lose the cilia and the mucus tends to build up within that individual's airway. So as we look at the cells that we use for differentiation, these cells are referred to as stem cells. And as we look at these stem cells, we can just loosely define them as an undifferentiated kind of cell. There's different kinds of stem cells. We have pluripotent, induced pluripotent, we have multipotent, we have totipotent. There's all these different kinds of stem cells there, and all these different names just refer to different stages of differentiation. But the thing that all stem cells have in common is they have the potential to differentiate. into one or more different kinds of functional cells. And this gives us some plasticity. This gives us the ability to flex a little bit during our development. So one kind of cell can form a variety, a diverse array of different kinds of mature tissue types. So as we look at these stem cells, we're going to start off with embryonic stem cells. And when most people think of embryonic stem cells, think of pluripotent stem cells, because these are the ones that are going to form cells of the embryo, so the future human being. But they do not form accessory organs of pregnancy. And you're probably thinking to yourself, what on earth is an accessory organ of pregnancy? I mentioned them again up here, these accessory organs that are made by the totipotent stem cells. When we think of accessory organs, I want you to think of things like the amniotic sac or the placenta, the things that the child needs to stay alive in utero, but that the child isn't necessarily going to be walking around with after the child is born. We then have adult stem cells. These are oftentimes just referred to as multipotent stem cells, and you as adults have a lot of adult stem cells. And as you age, as you add more decades to your life, the number of multipotent stem cells you have in reserve does decline. Some of our adult stem cells are going to be unipotent, so that they can give only one kind of cell type. For example, we have cells within that line, the seminiferous tubules of the testicles, that are only going to make sperm. And they do one thing: they make sperm, and they make lots of sperm. But we have lots, that's kind of the exception to the trend. Most adult stem cells are going to be multipotent stem cells. So as we look at my personal favorite, induced pluripotent stem cells, these are stem cells that are harvested from an adult or from somebody who's been born. And then after these cells have been harvested, they can be reprogrammed to mimic a pluripotent stem cell, to mimic a quote embryonic stem cell. And I get excited about this because now you have the ability to harvest cells from a human being and then reprogram those cells to be anything else within the human being. And you have perfect genetic compatibility. In addition to that, you're also going to bypass a lot of the ethical considerations that are associated with harvesting pluripotent stem cells. Because to get pluripotent stem cells, you traditionally have had to either destroy a human embryo or use cells that came from a destroyed human embryo. So as we look at these induced pluripotent stem cells, they're pretty cool. They've been around since, I want to say, 2008 is when induced pluripotent stem cells first hit the scene. And since then, some emerging therapies have been developed for stem cell treatments. Now, if we look at the tissues in our body, we need a way to repair those tissues. And we can do that two different ways. We can regenerate them, or we can have a fibrosis. And when we think of regeneration, I want you to think of replacing with something that's like brand new. So I have an old car, so I'm going to use some car analogies. When I need to fix something on my car, I can either replace it with something that is guaranteed by the manufacturer to be the same part, to have the same function. It can be as good as new. It's going to restore normal function. And this happens all the time. So, for instance, if we have a minor skin abrasion, we can repair that no problem. Or if we have a minor injury to our liver, again, we can repair that no problem. However, we don't always have the ability to restore normal function. So, going back to my old car, sometimes, for instance, I have a crack in my bumper. My rear bumper, I backed up into a tree. No biggie. And instead of spending several $1,000s to replace a cracked bumper on a car that's worth several $1,000s, I used duct tape. It got the job done. It works. It's not as good as it was before, but it's still, it got the job. It got the job done. We got her done. And that's what we can think of with fibrosis. So instead of replacing damaged cells with a similar or the same cell type, we just replace them with scar tissue. And the scar tissue is kind of like your body's version of duct tape. It just holds stuff together. It doesn't really have any specific functions other than just holding stuff together. So if you have any severe cuts or burns, or maybe if you've had tuberculosis, you could have some scarring forming. Most of us are typically going to have the severe cuts leading to scarring. Hopefully you haven't experienced severe burns. It's not fun if you have. But there are different things that can lead to fibrosis. As we age, our ability to regenerate tissues declines and our rate of fibrosis is going to increase. So when you look at young kids and they get a cut, you usually don't have to worry about the kid having a scar. But if you look at an older adult and that older adult gets a cut, then yeah, there's going to be a scar that stays pretty much for the remainder of that adult's lifespan. So as we're looking at, just a generic cut. So let's say, you're chopping, that looks like some green onions or maybe some green bell peppers right there. This lady's chopping up some peppers and she nicks her finger and she gets a cut. The very first thing that's going to happen is that blood is going to ooze into that cut. And within that cut, we're going to have a special kind of white blood cell called a mast cell, release histamine. And the histamine is going to cause the blood vessels nearby to become more permeable and dilate. And that's going to cause more blood plasma to ooze into the surrounding tissues. And that will carry antibodies and clotting proteins. The antibodies help to fight off microbial infection, and the clotting proteins help to initiate the blood clotting cascade. After that blood clot has formed, we then need to start to knit the edges of our wound together. And the good thing about these blood clots is that they make it very difficult for pathogens to get into our bodies because they have a very high density, a very high media density, so to speak. So because they're so dense, bacteria on the surface of our skin and on the outside of our body have to work very hard to get through. And this blood clot will ultimately form a scab that will block the wound site from infection and seal our wound. And then within the wound, we have lots of macrophages. And here's the diagram right there for macrophage. When we think macro, think big. Phage means to eat. Macrophages phagocytize debris within the wound site. So you could think of the macrophage as kind of like the cleanup crew. And this cleanup crew, this macrophage, is going to troll and roam through the blood clot and the scab and start to clean up site of injury. After a few days, we're going to have new capillaries growing and penetrating into the site of injury, and those deeper portions of the blood clot will also have fibroblast generating more collagen fibers. So we're going to have a soft mass of tissue known as granulation tissue. And then as the macrophages continue to move upwards throughout this growing tissue mass, they're eventually going to start to remove the blood clots, and the fibroblasts are going to deposit increasing amounts of collagen. This process can last up to 2 weeks, but depending on the severity type, maybe it could be as little as 3 or 4 days. So I'm looking at my own finger right now. I cut it with a razor blade a couple days ago, and it's already starting to heal. Thank goodness. Because it stings every time I get juice in it. Now, after this process of fibroblasts generating those collagen fibers, the surrounding epithelial cells, particularly from the stratum basale of the epidermis, are going to grow into the site of injury and eventually restore that stratified squamous epithelium that makes up the epidermis. We have some tissue regeneration beneath the scab, and eventually it pops the scab off. The scar tissue that's deeper down may or may not show through the epithelium, but a lot of times you can feel it if you palpate or rub it with your fingers. And this phase can last up to 2 years. So sometimes it takes a lot of time for the wound to heal itself fully. Again, there's a constant healing process throughout this. Now, let's keep talking about tissues. We talked about tissue repair. Now let's talk about tissue death. There are different ways, different things that can contribute to tissue death. For instance, we have atrophy, which is going to be a shrinkage of a tissue through loss in size or number. So we have senile atrophy. That's just a part of the normal aging process. When we lose brain cells, we lose a couple muscle cells. We also are going to have disuse atrophy, or I like to call this couch potato atrophy. So this is a lifestyle induced atrophy. We also are going to experience necrosis potentially. Ooh, what the heck just happened there? So we get a necrosis potentially. And when we think of necrosis, this is a disease state, or we have a pathological issue or a disease issue, some kind of homeostatic imbalance. So there can be some kind of injury, there could be some toxins, there can be some kind of infection that can cause the tissue death. So atrophy is slow and relatively not painful, or relatively painless. Necrosis, though, is relatively rapid and painful. We can have an infarction contribute to a necrosis, and this is when we cut off blood supply. And if we don't have nutrients being delivered, the tissue dies. We also can have gangrene. And this isn't gangrene typically isn't from the blood supply being cut off, it's just from an insufficient blood supply. So a greatly reduced blood supply. So, for example, you could have a pressure sore, sometimes referred to as a cubitus ulcer. So, this is a form of gangrene that can happen if somebody, I like to think of it as extreme bleacher butt. So, somebody has spent a lot of time in one position and the pressure points in their body have been compressed for such a long time that blood flow has been reduced and tissues start to die. A lot of times we will see dry gangrene, and my wife works in a nursing home and sees this one all the time, where individuals are going to have a reduction in blood flow, and then they're going to start to have tissue death, particularly of their toes and of their feet. We can have wet gangrene, and this one, ooh, that one's scary. This is when we have liquefaction of internal organs. So this is the stuff of nightmares right here. If we have body tissue starting to dissolve, and this is typically going to be associated with infection. The one that comes to my mind is Ebola. And then we can also have gas gangrene. And when we think of gas gangrene, this is going to be typically from soil bacterium that will have hydrogen bubbles forming the tissues. Those hydrogen bubbles are going to get lodged and reduce the ability of blood to flow through capillaries and ultimately contribute to necrosis or tissue death. We also could have apoptosis occur. And when we think of apoptosis, this is generally speaking a good thing. It's a pre-programmed cell death. So our cells are going to die because we don't need them anymore, or they're going to die because we want them to die. We're telling it something in our body is telling them to die because they're no longer beneficial and potentially harmful for our body. And the best thing for us is to get rid of these cells. So common things that are going to go through apoptosis are precancerous cells in our body will have apoptosis occur. Cells infected with viruses will go through apoptosis, or cells, structures which we no longer need will go through apoptosis. And a lot of that last one, getting rid of structures we don't need, that typically is going to happen in utero or immediately after birth. Every single day, we have billions of cells in our body die by apoptosis. This is like the suicide switch, the suicide self-destruct program of our cells. And what we find is that there's going to be a signal on the extracellular, on the outside of the plasma membrane, and it's going to bind. And this signal is going to be called FAS. And this FAS will activate some enzymes. There'll be a cascade of enzymes being activated. Ultimately, at the end, we'll have an endonuclease. Endo meaning in, nuclease meaning nucleotide enzyme. Remember, ASE is for enzyme. So these nucleases are then going to start chopping up the nucleotides, the DNA within our body. And then we're also going to have proteases released. Proteases are enzymes that destroy proteins. And if we destroy our cells' DNA and we destroy our cells' proteins, then our cells are no longer with us. They don't make it anymore. And we have that cell death. That's all we have for this recording. If you have any questions or comments, please feel free to post them in our class discussion board. And as always, happy studies.